This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 161, recorded December 9th, 2011. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Hello, Vincent. How are you today?
I'm well, and you?
I'm just dandy. This is great. I'm in great shape.
All right, up here, Rich. It's a beautiful day up here.
Oh, good.
How about down there?
We got, I'm looking at cloudy skies here. 71 degrees. That's good.
71. It's -2 here, centigrade.
Are you kidding me?
For all of our centigrade utilizing listeners, -2 centigrade.
Well, I'm at 21 or 22.
We're into winter, but blue skies, no clouds, really nice day.
Excellent.
Also joining us today from Western Massachusetts, Alan Dove.
Pleasure to be here.
Good to have you as well. And you, how are you?
Oh, doing well.
Very well. Good to hear it. Dixon Despommier is down in the Bahamas.
Oh, his weather's probably pretty good.
Fishing.
All right.
I don't know why.
He's doing better than us, then.
I don't want to be fishing in the Bahamas. I want to be right here doing TWiV. This is what I want to do at this moment.
All right.
Excellent.
We also have a special guest today. He is from several places. New York University Medical Center, Rockefeller University, soon to be at the Whitehead Institute up at MIT in Cambridge, MA. Gabriel, Victora.
Hi, Vince. Thanks for having me again.
Welcome back.
I could really get used to this.
It's a nice thing to do. I agree.
Yeah, it's fun.
Turn your volume up there a little bit.
Okay.
Gabriel was here not too long ago when we talked about Ralph Steinman. And if you remember, we decided we were going to talk about antibodies and B cells with Gabriel. So here you are. We're going to do that.
We've said for a while we need to do more immunology here. This is really hardcore.
Yeah,
this is going to be immunology.
This will be good. But before we do that, I want to mention 2 public service announcements. First, please take the listener survey at triplemojo.com/TWiV. I know some of you haven't done it, so I know you're busy, but when you have a chance, take that.
I actually took it.
You did?
Yes. Takes a little while, but it's worth it.
How long did it take you?
5 minutes.
Oh, it's not bad,
not that
right?
bad.
A lot of questions, right?
How many people responding do we have now?
About 650.
Okay, we need what?
I want 1,000
more.
Yeah. I think that would be good. Then we'll stop bugging you.
That's only 50 since last week. Come on.
Someone told me the problem is they listen in the car, and then by the time they get where they're going, they've forgotten about filling in the survey.
Yeah, that's the same excuse people use not to give money to NPR. It's not cutting it.
Okay.
All right.
You heard it. You heard it from Rich. Fill out the survey. The second thing is ASM asked me to tell everyone about their collaborative filtering experiment. We are crowdsourcing a session at the general meeting of the American Society for Microbiology in 2012. So we'll provide a link to a website, Submit, Vote, and Speak. You can suggest a session. The crowd votes on it, and the top 5 vote-getters, I believe, will become a session, and whoever put them up will get some travel subsidy for the meeting. So right now, the top vote-getter is Putting Freshman Biology Under the Microscope. 5 heresies about microbiology every freshman biology student should know. So that would be a talk. That's the idea. Second one is crowdsourcing genome analysis with wikis and Twitter for rapid response. It's a lot of crowd-related stuff. I guess that's what you get.
Well, you're asking the crowd to vote.
Right.
It's very interesting. There's no real focused stuff. I have to put one up. I'm going to put one on the end game for polio eradication. How's that sound, Adam?
Good idea.
That's good.
Get some science. Or which is better, STS-246? Oh, no, I shouldn't do that one. Okay.
You can do that one.
That would be funny, wouldn't it?
Yeah.
Nobody would vote because no one would know what it is. All right, so those are my two announcements. Let's move on to science. So we thought we'd talk about antibodies, how they're made, B cells, and all that sort of thing with Gabriel, because that's what you like to do, right, Gabriel? You like antibodies.
Yeah, that's my life.
That's your life.
Wait a
They're-
minute. I
they're
have a
great
question
molecules.
before we start that.
Yeah.
Okay?
We want to
Okay.
know.
Because
We want to.
you used to have another life, right? You were a professional pianist.
Yeah, that was my former life.
And we've got this YouTube video of you playing, right?
Oh, you found that?
It's easy to find.
I mean, it's pretty amazing. So why the switch? What happened?
That's a good question that I get asked quite a bit. I don't really know. There are a lot of things, but mostly I think that I was a little bit tired of practicing piano all day long.
When did you start playing piano? At what age? Like -4?
Something like that. No, actually,
Haploid. He was haploid.
my formal training started when I was 13.
Oh,
I played before that, but formally 13, and then I played professionally for many years, and at some point I got after college, and I got a little bit tired of just the whole routine of practicing the same thing over and over, and I thought, I want to do something that is different all the time, and I thought
Interesting.
science was a good candidate for that.
so you could have had a career in playing the piano. Is that right?
I guess I'm not sure how good of a career, but I did have a career for a while.
Do you still play?
About once every 6 months, more or less.
Is that right?
I don't have
Wow.
a piano, so the piano at Rockefeller is conveniently placed in the library, which means
Great.
I don't have much time to play on it. So
When you do play, do you have skills, or they're all gone?
I think they're deep down buried in the back of some part of my brain.
Right.
They'll come out eventually after a few hours of warming up.
So you thought, how old were you when you thought science might be the way to go for you?
I would say 24, 25. Yeah, something
And
like
so what did you do to get going in science? You were in Brazil at the time, right?
Yeah, I was living in São Paulo at the time. My father is an epidemiologist, and I was talking to him about this. I said, I want to be a scientist. And he said, well, I have a very good friend who was a high school friend and also a med school colleague that has a big immunology lab in São Paulo. Why don't you go talk to him? And so I did. I went and talked to him. He said, okay, come to the lab, be a voluntary for a bit. And I went there, and I really liked it. And so I sort of got into immunology kind of by accident, but I stayed there. So it was very interesting to me.
So that's where you got your PhD then, in São Paulo?
No, I got a master's there, and then I moved to NYU
I'm
and
sorry,
Rockefeller.
of course.
Yeah, for a PhD.
I knew that. So then why NYU and not any other place in the U.S.?
They're the ones that took me. They were willing to take a pianist with recommendation letters from people they didn't know, which is not, I guess, common to all graduate programs.
Good for them to do that, right?
Well, they obviously made a good move.
Yeah.
Yeah.
Yeah, it turned out okay. I think it's a very good program. I learned a lot there, and I learned a lot also at Rockefeller by being in a very competitive lab there. So it was a very good combination.
So you wanted to do this so that you'd have more variety. Has that goal been achieved?
Definitely, yeah.
Yeah.
Definitely. Every day I do.
That's the cool thing
every
about
day I do
science.
something
You
different.
learn
yeah.
it all the time. Everything is different.
I mean, you do the same thing over and over, but you interact with people all the time. You exchange ideas, and for some months you're doing a lot of imaging. The next month you're doing flow cytometry. The next month you're doing ELISAs and making protein and cloning.
And lots of times, even if the tasks are the same, what's happening in your head is different.
Right, you're
because
doing that
evolves.
routine work, and you're listening to TWiV and having good ideas at the same time.
Hopefully what's happening in your head is different. I don't know.
Yeah.
There's some people where I think
And you're
it's
filling
always
out
the
the
same.
listener survey.
Yeah, right.
Yeah.
Right.
So when you went to NYU, this is Department of Pathology, right?
Right.
And how did you start this collaboration with Rockefeller and the Nussenzweig lab?
So I went to Mike Dustin's lab which is a lab that has really a lot of expertise in imaging the immune system, and it's really one of the top immunology imaging labs that there are. And they had this collaboration going on with Michel Nussenzweig at Rockefeller on B cells and germinal centers, and they were imaging for the first time germinal centers in vivo in living mice. And one of the things that actually got me hooked to immunology was exactly the germinal center reaction, how the antibodies have real-time Darwinian evolution happening within you in a week or two. I thought this was really cool, and then when I had the opportunity to actually work on this in a lab that I knew about, also Rockefeller, the Nussenzweig lab at Rockefeller, I thought this would be a very good idea. So I ended up working jointly in a collaboration between the Dustin lab at NYU and the Nussenzweig lab at Rockefeller.
And now you're getting ready to go to the Whitehead and start your own lab.
Yeah, that's right.
And you're going to continue the same kind of work, I presume.
Yeah, I love germinal centers, so
and
I'm going to stick to them for a while.
At some point today, we'll have to have you define what a germinal center is, but I think we need to start at the beginning, at the very beginning. Right, Rich?
Yeah.
So immunity is important for the stuff we talk about, viruses. So we thought today we would focus on the antibodies and the B cells, which is perfect because you work on them. And, of course, antibodies are important for preventing many virus infections and also for resolving some of them. It depends on the virus. But when you're first infected, you have an innate response, which is the earliest response, right? And then that's important for either clearing the pathogen or saying, I can't handle this. We have to hand it off to the adaptive response.
And that's the innate response. We've talked, a lot of our conversation about immunity is focused on the innate response. And that is non-specific from the point of view that it doesn't know much about what sort of pathogen it is.
Right. It only identifies non-self and says...
Right.
A lot of it happens within the cell that is infected. So I would think that virologists would like that very much because it actually it's actually a mixture of immunology and virology because of all the interactions that the viral proteins have with innate sensors.
Right, and it's clear that there is an evolutionary tug-of-war going on where the cell is responding and the virus is fighting back and blah, blah, blah. The viruses are developing countermeasures and the cells are developing more antiviral stuff. And a lot of this involves interferon pathways and stuff just to sort of ring a couple of bells.
Of course, the dendritic cells that Ralph Steinman found that we talked about last time Gabriel was here.
All kinds of signaling pathways and everything else. And there's no memory, right? This happens at wane. You get an innate immune response. Eventually it wanes, and it doesn't remember that anything happened relative to this pathogen.
Exactly.
So, Gabriel, let's say you have a virus and it overwhelms the innate response. So some infections are cleared by the innate response and never go beyond that. But if it goes beyond a certain level, you have to engage the adaptive system, which includes the antibody response and the T-cell response. And so today we want to talk about the antibodies. So what is the earliest thing that happens in one of these preceding viral infections that would start antibody production?
Well, there are two things that happen pretty early on. I would think if you focus exclusively on antibodies, a lot of viruses are very good at eliciting what's called a T-independent B-cell response. And the definition of this is operational. It's a response that can happen in a mouse that doesn't have any T cells. Mm-hmm. So some mice that don't have T cells can respond very well and with a very large magnitude response to certain types of antigens.
But not all, right?
Not all types of antigens. What's common about these antigens is usually that their conformation is in an array. So there are several repetitive determinants of these antigens. Now, these kinds of antigens, what they do is I guess I have to go back a little bit to what an antibody is.
Yeah.
Right.
Sure.
So what an antibody is it's a protein molecule. And it's a Y-shaped molecule. And what what it does is that you imagine a Y, and imagine that the two tips of the Y that point upwards actually bind to antigen and stick to stuff. So that's what antibodies do. They stick to stuff that's hanging around in the serum. The bottom part of the Y is what's called the constant region, and it's the same for all antibodies. It doesn't matter if they stick to smallpox or to red blood cells or to a chemical, little chemical group. It's always the same. It's called the constant region. And that has many forms, but basically the molecule is divided into roughly two major forms, one of which is soluble in the serum, and the other one which is attached to the surface of a cell called a B cell. And what a B cell does is it has these molecules attached to its surface. When it gets a signal saying, okay, this antibody is important, they differentiate into this cell called a plasma cell that just makes the other version, the soluble version, and spits it out into your bloodstream. So the B cell has the sensor, which is the antibody on the surface. It gets activated, it becomes a plasma cell, and starts to spit it out in the serum.
So and each B cell makes an antibody against a different antigen.
Exactly. So the B cells, we can go into how they diversify their antibodies later, but each B cell has a different receptor that could be specific for something important, such as a virus, but it could also be specific for nothing. Could just
and you're born with a library of these things, basically, right?
You're constantly generating this library.
Okay.
I am even now generating libraries of.
You're generating many millions of B cells.
So at any given time, how many different specificities do we have?
Oh, that's a good question. Something like 10 to the 11th.
Wow.
Or that's maybe an overestimate.
So we have that many cells, and where are these cells with these antibodies on them? In the blood? And
they're in the blood, they're in the lymph nodes, a whole lot of them are in the spleen.
So they're doing surveillance. They're running around looking for something that matches the antibody that's sticking to their surface.
Yeah, they spend a little while in the spleen, then they get out into the blood, they go to a lymph node, spend a little while there. And
so if I get a rhinovirus infection, then by chance, eventually, the virus will encounter one of these cells with the right antibody on it. Is that how it works?
Yeah, that's right. So that's what I wanted to get down before I go into the T-independent response. Mm-hmm. So what's important is to know that the antibody exists in two forms, a cell-bound on the B cell and a soluble form in the serum. And the soluble form is the active form that will actually fight the pathogen.
So for every cell-bound type of specific antibody, there's a soluble form.
Not at the same time. Usually, you need the B cell to get activated and then become a secreting cell.
All right. So the first thing is for this B cell with the antibody to interact with the pathogen.
And one way
interaction between the antigen and the antibody activates the cell, and it divides and then starts making this.
Exactly.
Starts making the soluble.
And now we go back to what you asked about what is the first response. So the first thing that happens is there are certain viruses that have these arrays of antigens on their surface that are spaced at a certain distance from each other, something like 5 to 10 microns. And if they have enough of these determinants, a large enough array of these determinants, and they stick to a B cell, what's going to happen is this B cell is going to say, whoa, this is dangerous. Let me transform into a plasma cell and make a lot of this antibody. And this is independent of a T cell being there or not. So it's very fast.
Within
a few days. So these B cells are usually not class switched or maybe very little of it. So they make this type of antibody known as IgM. So the first thing that happens when you get in contact with many types of viruses is this early IgM response, which is not dependent of T cells. And that is often it's often sufficient to at least, if not clear, hold back the infection until another more mature response comes in.
So what do you mean by IgM in classes?
Okay, that's another issue. So the antibodies...
Immunology is just great.
Yeah, it's kind of hard. You have to go backtrack to everything. So I told you that the tips of the Y varied a lot, right, between different B cells, and they could be specific for any type of pathogen or anything, and that the bottom part was more constant. It's not completely constant. There are 5 or maybe 7 different types of that stem. And depending on what kind of stem the antibody molecule has, it's going to have a different function in the body. The first type that all B cells have before they are exposed to antigen is this type called IgM. And this stem, what's important about it is that when these molecules are synthesized by the B cell and exported into the bloodstream, they form groups of 5 molecules, in which we call it's a pentameric IgM. This means that even if the antibodies are not very good against the viruses, they can still stick pretty well, because it's like 5 Ys stuck together, and it means they have 10 tips that can bind to the virus. So actually what you get is, instead of 1 or 2 sticky tips, you have 10, and that makes it very easy to bind to the virus. Another thing that's particular about IgM is that it is very good at recruiting this pathway that we call the complement pathway, which is in the serum. So it's the antibody can stick to a virus, and then complement's going to stick to that antibody, and it's going to coat the virus with complement, which is very attractive to certain types of cells, the phagocytes, like the macrophages, and etc. And so when a virus is covered up with complement, the phagocytes will say, well, this is something that I should eat. And they'll scavenge all the complement, all the viruses that are covered in complement, and eat them and digest them into little peptides, and then they're not dangerous anymore. So that's IgM.
So the process effectively flags all these things that wouldn't normally be recognized by a lot of cells, flags them as danger.
Right. It's like putting a tag on something and say, this is trash.
Right.
Remove, please. Right.
Is that what we call opsonization?
Exactly. It's called opsonization by complement. Okay. But IgM itself is not very good for opsonization. What happens is sometimes after a B cell is activated, it can switch the stem of the Y into other different classes. And one of the classes is IgG, which is very good at opsonization directly. It doesn't need the complement to bind. So if there's an IgG bound to something, a macrophage will go there and eat it, regardless of whether there's complement on it. The antibodies can also change to other chains, such as IgA. IgA is present in secretion, so it's present in the gut in very large quantities, and it's also present in the saliva and tears and in the lung mucosa. And one of the principal characteristics of IgA is that it can cross these epithelial barriers and be present in high concentrations in extra body fluids.
But I don't know if this is the right time, but for IgA, the B cells with IgA, do they also cross, or are they always in circulation only?
The B cells are usually inside the lamina propria of the mucosa. That's where you can find most of them.
So the virus has to get down there.
Or the Ig has to get out.
Well, but you said that to amplify the specific antibody, the virus would have to contact this.
Right. It will contact the cell before it's an IgA cell. It will contact it.
Okay.
So we're sort of getting ahead of ourselves.
Yeah.
And is this getting too complicated?
No, it's
beautiful.
fine.
I love it.
Yeah, it's great.
We're sort of diverging. But
no, okay. That's okay. That's what we do.
We'll get back to...
You're very clear. You're doing a good job.
It's really good.
We were doing IgA.
We were doing IgA.
We were kind of done with IgA.
So we had IgM, IgG.
We were doing class switching.
IgA, and there's another one.
So there's
IgE.
different stems
IgE.
are the classes. So when you're talking about class switching, it's taking a specificity and switching the stem on it so that it has a different function. Right.
It's 2 parts, right? The specificity determining part and the effector function determining part.
Right. Okay.
So an antibody has to do 2 things. It has to stick to something, and it has to tell the system to do something about that.
Right.
Right.
Okay.
So the class switching is taking the top part of the Y, which is specificity determining, and just swapping it onto a different base, which determines a new function.
Exactly.
Okay.
So the last form is IgE, which is infamous for causing allergies. And it's very good at degranulating these cells that we have that are called mast cells. So these mast cells are degranulated. They release all sorts of really noxious chemicals that can give us allergies and make huge inflammatory reactions. It's thought that the reason for this IgE to exist is to fight off helminths and other types of intestinal parasites, because a mast cell or a basophil or a eosinophil would stick to the parasite and release all sorts of noxious chemicals into the surface of the parasite, which would lead to expulsion.
Do viruses induce IgE, or usually not?
Viruses usually don't.
And it just stops at IgE.
That I know
right?
of. Yeah, it's usually IgG. It depends on what route they are introduced.
So when in clinical microbiology, often if you're looking for viral antibodies in a person and you find IgM, that tells you it's early in the infection. And if you find IgG, then it's much later.
It means it's later.
Right, for the reasons that you've just talked about.
Yeah. So back to where we were. So the first thing that happens is this T-independent response to these determinants that makes the B cells go into antibody production mode without, I guess, thinking too much about it.
Very good.
Now, the other thing that will happen, is that dendritic cells, which are the cells that we were talking about last time I was here, that were discovered by Ralph Steinman, they will capture these viruses, they will process them, and they will present them on their cell surface to another type of lymphocyte that's called a T cell. Now, a T cell is very much like a B cell. There is a huge population of T cells, and they each has a different receptor on their surface, which is randomly generated or quasi-randomly generated, and that can respond to anything that may or may not exist. Okay, these cells live in the lymph node.
The T cells.
The T cells. Mm-hmm. The dendritic cells live in the mucosal linings and in the skin and all sorts of other places. When they find a virus and eat it, they get activated, and then they migrate into the lymphoid organs, such as the lymph nodes and the spleen. And
this all just blows my mind, you know? Every time this just blows my mind. I got all this stuff going on inside me.
Yeah, well, and we can see it happening in real time. With these new microscopy
Sorry,
techniques.
I just had to go crazy there for a minute.
So these are, they are sentinels. They're out there. They're waiting for something weird. When they see something fishy, they take a piece of it and go into the police station and start showing pieces of it to the T cells. And the T cells...
I all of a sudden had an image of a dog fetching a newspaper.
It's something like that. And the T cells, if a T cell that can recognize that thing that's being presented by the B cell happens to be around, so a T cell whose randomly generated receptor will stick to the surface of a dendritic cell presenting a piece of a virus, that T cell will start to expand. And these T cells expand.
You mean divide, right?
Divide, right. Mm-hmm. They divide many times and they become a large population of T cells. Mm-hmm. I'm not going to go into the effects that the T cells have alone on the virus, which would take another whole episode. But I'm focusing on the B cells. What happens is, at some point, you have enough T cells that they are all over this lymph node, okay, or spleen. At the same time, certain B cells got activated by little proteins that are not in arrays. So some viruses don't have these arrays of proteins, like HIV, for example. So the B cell won't go into antibody production mode immediately. It will take a little piece and say, well, this is something I can stick to. Let me go see if there's a T cell that is specific for this virus as well. And if I find this T cell, then I will go into antibody production mode. Okay, I'm anthropomorphizing this to an extreme, but
that's good.
I think that's the
Why does it
idea.
wait for the T cell?
That's a good question. It's, in a way, I think it's something like a double lock system to prevent cells from reacting to self components.
Yeah, self is a real problem.
So imagine that a B cell happens to think that albumin in your serum is something it could stick to. It's going to go in and try to find a T cell that's specific for albumin. It's not going to find it, so the B cell is just going to die and you're fine. So you have to have two cell populations authorizing this to happen. And it's really funny, if you allow me to go in a little
It's sort of like the
site.
two guys turning the keys on the missile launch
Right,
panel.
exactly.
Yeah,
That's my analogy.
Mm-hmm.
Other different systems, like the lamprey, that has a sort of a peculiar primitive immune system that's not related to our immune system, also have this. They have a soluble component and they have a cellular component, and they also have this double key system that evolved completely separately from our own jawed vertebrate system. And it's really interesting that in two systems that evolved separately, you have the same thing where two cells have to authorize the production of these humoral, these soluble things, antibodies in serum or antibody-like molecules.
Well, then you could ask another. Then you could ask the converse question. Then, and why then is there T cell independent B cell production? Maybe those viruses are just so nasty that we'd better just do this right away without checking, or something like that.
Well, I think there is a double key system there, but the double key is operating on the same cell. So one thing is I can stick to this. The other key is it's arrayed in a way that I would never detect something like this inside my own body.
Okay.
So there are two pieces of information.
I see. Okay, fine.
Okay, one is I stick to it. The other one it's arrayed.
Okay.
Well, but that's a hypothesis. I don't know if that's
Good enough for me.
true or not. But
So just to review so far, so when the original B cell with the antibody on it interacted with an antigen, it comes back into the lymph node?
the B cell?
Yeah.
The B cell's already in the lymph node.
How does it get the antigen?
The dendritic
from
cell
the
went
dendritic
there.
cell?
Actually, the antigen comes into the lymph node in two ways. It comes in via lymph draining.
Mm-hmm.
So the lymph node is a node that is on a lymphatic. The lymphatics drain the fluid that comes from our tissues
Right.
into the lymph nodes. So, for example, if you have if you step on a nail, okay, you get all sorts of innate immunity signals in your foot.
Mm-hmm.
Your foot's going to get inflamed. It means your blood vessels are going to become leaky. So a lot of fluid is going to leak out from your blood vessels and it's going to drain back in via the lymph. So all sorts of pieces of bacteria or pieces of viruses that might have been on that nail are going to drain back, and your lymph node filters that. So at the same time, these dendritic cells that capture antigen are also migrating through lymph up into the node. So you have two waves of antigen. You have a early soluble wave that can activate the B cells, and then a later cell-bound wave that can activate the T cells.
All right, so the dendritic cells talk to the T cells, and then the
Right.
soluble antigen comes in for the B cells. Okay.
Now, what really puzzled immunologists for a very long time was how come you need a B cell and a T cell to be specific for the same thing? These are both 1 in a billion events, or say 1 in a million events.
Mm-hmm.
How do these cells find each other? And so, and now by looking at these cells in vivo with these microscopy things, we can actually, sort of figure out how these things are really happening. The dendritic cells come in, they talk to the T cells. The T cells proliferate. So one T cell that's specific for that determinant becomes 1,000 T cells that are specific for that. So the B cells migrate to the border between the place where the B cells stay and the place where the T cells stay in the lymph node. And all the T cells that are specific have also migrated to this place. So there is a place where the B cells can easily find a T cell that was exposed to the same determinant. And if that encounter takes place, then the B cell will expand and do several things, one of which is immediately differentiate into a cell that makes that antibody.
So there are compartments in the lymph node, but cells move among them.
Right. The B cells circulate in the follicles, which are the areas in the lymph node that are full of B cells, whereas the T cells circulate in the paracortex, which is also known as the T zone.
Mm-hmm. Sounds like a place where you play video games.
The T zone.
The T zone.
Yeah.
So the interfollicular space is where the T cells are, basically? Or is that what it's called?
The interfollicular space is more specifically the edge of the T zone that touches the capsule.
Okay.
But there's this compartment we call the TB border.
Right.
So once there's an immune response in process, the specific cells that have recognized the pathogen move to this TB border. and there they can meet each other and exchange the signals that lead to the B cell producing antibody.
So part of your work has been to look at this interaction in the lymph node, right, between T and B cells.
Right.
So maybe, which is we probably should wait to get to, right, before we should finish going through the antibody response.
I mean, I'm almost getting there, because one thing that's important about this process that I just told you is that, okay, let's see how I can go about this. There are, say, 1 billion B cells in your body, or maybe 10 billion.
Mm-hmm.
And these B cells are all specific for different things, but they're all generated randomly. So the number of substances out there that the B cells could recognize is much, much greater than 1 billion.
Mm-hmm.
Right? It's much greater than 1 trillion. It's almost an infinite number of different conformations of molecules that a B cell could potentially stick to. So you have a little problem there. There's much more variety of molecules out there than there are possible B cells in your body. What happens is that you actually don't get B cells that sticks very well to the stuff that comes in. You get B cells that stick sort of more or less well. Okay, and that's enough to trigger them to get started.
Ah. So for anything you might encounter, there's a B cell that at least will weakly interact with it.
And we call this a low affinity antibody.
Okay.
Okay, it will interact with it not very strongly, but strongly enough to get the B cell started. Now, if you look at antibodies that are produced during the immune response, the antibodies that are produced early on, at about a week or so after the beginning of the response, they're all low affinity. As the response progresses, these antibodies become better and better at sticking to these things. And then finally, after maybe a month after the original immunization, you have this population of molecules that we call the high affinity antibodies, which stick to the virus at a rate that sometimes is up to 10,000 times better than the original. And this process was discovered by Niels Jerne, who got a Nobel Prize for monoclonal antibodies. And it was also shown very nicely by Hermann Eisen, who's a researcher at MIT now. And they named this process affinity maturation of antibodies. And this is what I study. And it's really interesting because there seems to be some sort of education of the B cells to produce better and better antibodies. And that is very, the mechanism by which that would happen have puzzled people for a very long time.
Right. So what's going on is very much like I think we've talked about high throughput screening in the pharma industry. And so you get some initial hit that sort of works, and then you try to improve it over time. And the question is, how does that happen?
How does that happen? How can we actually do this within our bodies in a relatively short period of time? And the answer to this, which has become clear in many decades of studies, is that while some of the B cells that are activated by T cells early on go straight into the antibody production mode and are responsible for this early wave of low affinity antibody, other cells go back to the center of the follicle and the B cell zone where they were hanging out to begin with and start to proliferate there. So one B cell becomes 2, becomes 4 and 8, and it's they ultimately form these structures that are called the germinal centers, which are structures within the B cell zone where a lot of cells divide very rapidly. Now, what's really interesting about these cells is that they start to edit their DNA at specific places. More specifically, the antibody receptor gene, so the B cell receptor gene, which dubs as an antibody after it's secreted by the cell. And this is done by this enzyme called AID. It's an activation-induced cytidine deaminase, known as AID. And what this enzyme does is, in a pretty well-targeted manner, it starts to introduce little lesions into the DNA right at the part of the antibody molecule that binds to foreign substances. And this is a random process, so you could think that out of 100 mutations that this AID enzyme introduces into that part of the antibody, 99 are going to be bad. Some are going to create stop codons, and the B cell receptor is going to stop working. Some are going to make mutations that the cell doesn't care about. Some are going to make mutations that makes the cell worse. And what happens is somehow inside the germinal center, the cell gets selected. So it's very much like Darwinian evolution. There is a generation of descendants with modification and then a selection process
for the high affinity.
that purges the low affinity cells or the lower affinity cells and keeps the best cells around.
How does AID know to just mess with the antibody combining site region?
Oh, that's the million-dollar question. And I think we're getting close to some really good clues as to how that happens. Apparently, stalling of RNA polymerase has a lot to do with this.
I love to hear it.
So that's what my lab is working on right now, and I'm not the expert on it in the lab. But
right. Who discovered AID?
It's Hanjo. It's
Takashi Hanjo.
Yeah,
it's an important discovery,
in Japan.
right?
It's a very important discovery. It opened up a whole new field that is probably the hottest field in B-cell biology.
So you might get a Nobel Prize someday, right?
Well, that would not be unwarranted. And, well, it's sort of not known exactly how the targeting works, but this molecule, SPT5, has been shown by our lab to be important for bringing AID to the polymerase complex.
Ah.
And SPT5 is associated with polymerase that's stalled. So we know, at least for the some regions of the B-cell receptor where AID targets that are not necessarily the mutation, but something that's just to do with switching the class isotypes, there are lots of repetitive regions. The polymerase has trouble getting through those regions. And because of that, it gets stalled there, and then AID comes in. It exposes single-stranded DNA, which is the substrate of AID. And AID can go there and deaminate cytidines into uracils, which get recognized by mismatch repair systems and et cetera, and lead to either editing of DNA with point mutations or double-strand breaks.
Wow.
That's
just great.
It's really an amazing...
It makes perfect sense
an amazing
to
system.
Use RNA polymerase, which is not only stalling, but it's separating the strands. And
right, so the strands are melted. And,
wow.
Yeah, but the details are still coming out at a very fast pace. So it's a very exciting area right now.
So this contributes to the production of high-affinity antibodies, right?
It generates a panel of mutants upon which selection can act. Right.
Is there also a role for VDJ recombination in this? Or is that
VDJ
different?
has happened before, so there's no further VDJ.
And that gives you the different classes?
VDJ recombination gives you the different specificities to begin with.
So that gives you that initial panel of a billion
we can get
different
into that
B-cells.
if you. We could go through that a little bit.
Just give us an executive summary.
Executive summary. How do you make a billion different antibody molecules if you have a genome that is finite?
Right.
The answer is you use combinatorial diversity. So each antibody gene, when it's mature, it has a V segment connected to a D segment connected to a J segment, and these segments are right at the tip of the Y. And these are the segments that bind to things that come into the body. Right? But there's actually, inside the antibody gene, there is something like, in the mouse, something like 150 V genes, about 10 or 12 D genes, and 4 J genes that can get combined in different combinations of, you can get V number 100 combined to D number 3 and J4, and et cetera, et cetera. That generates already a lot of diversity. And that's on one chain of the antibody, what's called the heavy chain. This also happens in the light chain, the kappa chain. It can also happen in the lambda chain to a lesser extent. So by combining all these possibilities, you can create a huge library.
I see. So the problem was that we had more antibody specificities than coding capacity in our genome.
Right.
And so this was the
solution.
So who got the Nobel Prize for that?
That was Susumu Tanagawa.
Okay.
Which was really, one of the major discoveries in immunology was this rearrangement.
And that's all happening at the level of DNA recombination during development, right?
That's during B-cell development, not during organism development.
Right, okay.
Right.
So am I still doing
VDJ?
Yeah, okay.
In your B cells, not so much in your T cells.
Okay. Yeah, I can feel
Actually, there's something I wanted to...
Sorry.
You guys were asking what NF-kappa B stands for in TWiP?
Yep.
Yeah, so NF nuclear factor for the kappa chain that binds the enhancer of the kappa chain of B cells.
Right.
That's what the kappa B means.
So it was thought to be a regulator of kappa gene
somehow,
expression. But I think it turned out not to be specific for that, right?
Right, it does all sorts of things.
Yeah.
But it's just the origin of the name.
It's 25 years old today. This year, I mean.
Yeah, there was a lot of...
The discovery, anyway.
Buzz about it. Okay, so where were we?
All right, so we got VDJ happens to give you that initial billions, and then somatic hypermutation gives you something to select.
Right.
Yeah, so now we have a library of maybe good guys, maybe bad guys. We've got to select out of that the good guys. After
a mutation, right.
And meanwhile, the virus is still replicating, right?
Right. Meanwhile, the virus is still there doing damage, and that's why it takes, well, maybe that's why it takes a week to get rid of a flu, because you need the...
it's a lot of work to be done.
Yeah.
You need clonal expansion and that stuff to come out.
Mm-hmm.
Yeah, you're developing a customized response against this thing. That
I'm actually not sure how much this process helps you get rid of a flu, but
yeah, in fact,
I think T cells are more important.
T cells may be. For some viruses, certainly the antibody response is important for clearing, but we typically think of antibodies to prevent new infections.
Right, vaccines. That's the reason why vaccines work is because of all these processes.
So we go through all this and more, which you're going to tell us, and in the end, it's not really very useful, right?
Yeah, that's what Rolf Zinkernagel always says when he says the concept of immune memory is useless because if you've survived the infection once, you're likely to survive it again. So why would there be memory?
But you wouldn't necessarily survive it the second time.
Yeah. Well, it's just I think he's just being
weird.
Facetious. Yes.
Yeah. So okay, and what I do, I work on is how exactly does this process of mutation and selection happen. So if you want to, if you want to talk about the work that I did, then I think now we have the background and we're there.
That's great. Yeah,
that seems like a good segue.
So the question I tried to address was how exactly is selection happening in the Germinal Center? And
so I'm sorry. How did we know that that's where selection occurs?
Okay, that came from work by many people. I think notably Klaus Rejewski and Garnet Kelso in the early 90s, late 80s, who started single picking cells from germinal centers and sequencing them.
Wow.
And they found that these cells had mutated genomes, whereas cells outside the germinal center didn't. They had the germline version.
And sequencing was not as easy then as it is now.
Especially from a single cell. So these were, tour de force works.
So this selection just occurs in lymph nodes or anywhere else in the body?
Germinal centers occur in lymph nodes. They occur in the spleen. We have the Peyer's patches in the gut.
Okay.
We have them abnormally in sites of heavy inflammation.
Okay.
We have them in tonsils. That's
right.
That's the main source of human germinal center cells for study are tonsil samples, specimens from people who have tonsillectomies.
So the way you describe it, it sounds like a given germinal center is represents in response to a pathogen, a novel clonal collection of B cells. Is that right?
Right. It's not necessarily a single clone. So a germinal center can be founded by a number of clones, but you generate a new library.
Okay.
So you have your old library that is at best low affinity, and then you generate a new library. Okay. Where new higher affinity antibodies can come
for the purpose of a specific response.
Right. Now, what's one of the important features of the germinal center is that it's divided into two parts. We could call a light zone and a dark zone. Okay. Now, the dark zone is just a mass of B cells that are dividing constantly and produce and mutating their genomes. Okay, this is what was thought to be going on. Once the B cells mutate their genomes in this light zone, they were thought to be exported. In the dark zone, they were thought to be exported to the light zone. Now, the light zone contains a lot of other cells. It contains these really beautiful looking cells that are called the follicular dendritic cells. They have nothing to do with the Ralph Steinman dendritic cells. They're just also dendritic, and that's why we call them that. They're called these FDCs, follicular dendritic cells, are antigen depots, basically. So they have receptors on their surface that can bind to immune complexes. And immune complexes are these early antibodies that your body makes that are not very good, stuck to viruses or pieces of viruses. They make these clumps, and these clumps get deposited on these cells and held there for very, very long periods of time, maybe over months and maybe even more. So these cells are basically a depot. So you'd think, well, the B cells are mutating their genomes, and then they need to be selected. So there needs to be somewhere where antigen is available so that they can try it out.
Wow.
I was going to ask about this because a lot of this sounded like it would go on for longer than the pathogen might even be around. But there's a depot.
It probably goes on much longer than the pathogens around.
But these cells hang on. These cells are, as you say, a depot of antigen. So I can keep that around even though the pathogen is gone. Wow.
Yeah, and it's very easy to label them for microscopy, for example. So if you just take an antigen that's fluorescent and make the mouse recognize this antigen by injecting antibodies against it into the mouse, you inject the fluorescent antigen, it's going to go and stick on the FDCs and label them beautifully. So they're super efficient at scavenging this antigen and keeping it on their surfaces. So the B cells move up and they meet this antigen on the FDCs. Now, in addition to the FDCs, there are also a bunch of T cells in the germinal center. And these T cells are also specific for the antigens that are driving the germinal center to begin with. And one of the things that B cells do is to eat up antigen on the FDCs, digest it, and then present it to the T cells. And these T cells can also give survival signals to the germinal center cells. So basically, the cells mutate, they generate all the different mutants, and then they go up and they talk to they see the antigen on FDCs, pick it up, talk to T cells, and somehow in the middle of this process they get selected and told to become antibody secreting cells and go into the bloodstream and make a lot of antibody. Or they're told to become memory cells that are just sort of quiescent B cells that hang around waiting for the virus to come again.
Do they stay in the lymph node, the memory cells?
They recirculate just like naive B cells. They go from lymph node to blood to spleen, etc.
And that's where the memory to the pathogen comes from. So that next time that's going to be that cell is encoding now an antibody that has gone through this maturation.
And that's already good, and is already in much higher numbers than its precursor would be.
So you can get a quicker, better response next time around.
And it's not just a little bit better. If you look at the plots of antibodies produced after a secondary challenge, it's about 2-3 logs higher than what you get in the primary.
And those are already high affinity.
Those are already high affinity.
They've been selected and stored in memory.
So you get a super fast response within 2 or 3 days, which is much, much higher than
it's brilliant.
Yeah, it's an amazing system. Now, what I started to work on was that there was this idea that, if you take B cells in the dark zone, they're constantly mutating and exporting cells up into the light zone. That was sort of known from radio labeling studies. Now, there was this idea that in order for this process to be efficient, you couldn't do it in a single round. So you couldn't expect cells that are mutating in the dark zone to all of a sudden put in the 5 good mutations and not put in any of the 10 potentially bad mutations. That would be a very low chance of that actually happening. So computational biology people came up with this model called Cyclic Re-Entry. They said that the cell would be in the dark zone, it would generate a bunch of mutants, it would go up to the light zone. If it was selected, with some probability, the cell would go back to the dark zone and do it again. And so the cells would be cycling up and down, and after several cycles of mutation and selection, just like in Darwinian evolution, you would get a much better antibody.
Wow.
Let me interrupt here for a minute. So the selection we're talking about is basically this interaction with the follicular dendritic cells and the T cells, and there has to be a match with the antigen, right?
Right.
And that's what selects those guys to hang around. The cells that have hypermutated so that they don't have a good match anymore, they can't bind the antigen, can't bind the FDCs, can't bind the T cells. So what happens to them?
These cells die.
They die.
They just die by apoptosis, and they get eaten by these macrophages that live in the germinal center as well.
All right.
Right,
So,
so
we
the
get
cells-
rid of the
the
bad
cell-
guys.
Right.
Yeah.
So the B cells hang out, they mutate, then they go out and take a test. And
Right, and if they fail, they die.
if they fail, they get fed to the macrophages.
Yeah,
They get
exactly.
and if they pass, they at least a few times around, they're told, "Okay, nice job. You go back and work on that some more."
Exactly. It's a nice thing to
Come
tell your
back out.
kids. If you don't
It's
study,
great.
you're going to get fed to the macrophages.
Hey, Rich, you remember we talked about the T cell receptors and HIV susceptibility?
Yeah,
Same
yeah.
idea. In maturation in the thymus, the T cells that bind too high affinity, they get killed.
They get killed.
Too
Right.
low affinity get killed, right?
Right.
Goldilocks, the ones that are just
There's
right.
a whole lot of violence in the immune system
Amazing. And this is happening
state, and you wouldn't want it any
It's
other way.
happening in your tonsil right now,
right now. I'm sitting here, and there's all this death.
and in your gut.
No wonder we feel bad.
I feel pretty good.
Okay.
Well, you must not be
I have
mutating,
something's
man.
wrong.
Okay, so where was I?
So the follicular dendritics and the T cells are interacting with the B cells,
And they're doing these cycles, right?
cycles back
of
and
testing,
forth,
right.
right?
But that was theoretical, and that was really hard to show, and that's where our work came in. We started, not me personally, but people in the lab before I came, started having a look at the germinal centers in vivo. And for this, we use this technique called two-photon microscopy.
Which is made of awesome.
It's a really neat system.
Explain it.
Please
I don't
describe
guess.
how this works.
Okay. So, do you want the physics? Probably not, right?
Not detail on the
Okay,
physics,
let's
but
skip
just
the physics.
the concept.
The concept is
Do you
that
know the physics?
barely
Okay.
enough to talk to the microscope guys and tell them to tweak this or that so that I can see better. But what happens with this microscope is that you can see things that are deep into tissue without doing any damage.
You don't have to cut the tissue.
You don't have to cut the tissue.
But you do have to take out a lymph node from the mouse, right?
You don't. You can do it inside the live mouse.
This
Are you
blows my
serious?
mind. So what do you got, a leg
I
sitting on
have
the
a
stage
picture
or
for
something?
that. Maybe I can give it to you later. No, what you do is this. So this laser allows you to see inside tissue down to, say, 300-400 microns, which is enough for us. Now, what you do is you take a live mouse, you anesthetize it with isoflurane. This is all very done in accordance to all the ethical rules that we have for dealing with mice. When the mouse is anesthetized, you do a tiny little microsurgery in the back of the knee. That's where we image. Other people image other sites. And you open a little slot in the back of the knee, and by microdissection and under a stereoscope, you carefully expose a piece of the capsule of the lymph node. Okay. Now you take this mouse with the exposed lymph node, you put it under the two-photon microscope, and then you can start the microscope while the mouse is still alive, and you can see the cells moving around. So all these things I told you about, cells moving in and out around the follicle and moving to the border and moving back, all these things have been documented by two-photon microscopy.
And you can actually, with the two-photon microscope, right, you can actually sort of do visual slices through this at different layers, right?
Right, you can look in 3D. So you get these beautiful 3D images of structures. For example, the germinal center is easily within reach of the microscope, so you can actually make a 3D model of a germinal center.
Why is it two-photon? What does that mean?
Two-photon means that two photons have to hit the same fluorophore at the same time so that it will emit
I
light.
see.
And
So-
that only happens at the focal point.
Right.
That's why you can go deep into the tissue without getting light from above or from below, because the two-photon effect happens only at the focal plane.
Right. And the resolution on this allows you to look down at the individual cells and exactly what
Right.
they're
It's
doing.
not super high resolution, but it's enough that you can clearly see cells, and you can also see little pieces of cells and vacuoles and all sorts of stuff.
Okay.
So
These are very expensive at microscopes, right?
they are. They're getting better now. Companies are now making them. When we began, we had to all do it by hand.
Okay.
I'm picturing a mouse hanging off the stage here. Okay, is this Have I got this? I need to see a picture of this in action.
Okay. Let me see if I can describe it. You have a little heated stage that's like a bed. It keeps the mouse warm because anesthetized mice tend to go cold. It keeps the mouse warm. It has a little anesthesia. We use isoflurane, which is a gas, so there's an anesthesia mask attached to this, and the head of the mouse is inside the mask. And the mouse is attached to the stage with a little sort of a strap over the leg right where the lymph node is, and that sort of keeps it in place. So the mouse is lying on the stage under the objective of the microscope.
How much time do you have to do the visualization? Do you have minutes or seconds?
No, you can do hours, many
Really?
hours, yeah. We have time courses of 10 hours.
And the mice are fine?
They're fine. The isoflurane is great. They do surgeries on veterinary surgeries are all done with isoflurane, and they can go on for a long time. So
okay, great.
So you can watch individual cells moving.
Right. So that was the first thing
around.
people wanted to do. Let's see if the cells go from the dark zone to the light zone and from the light zone to the dark zone.
Right.
Okay, but there's one limitation to this, that you can't make very large movies of the whole lymph node at a fast enough rate that allow you to track the cells. Because it takes about a minute, no, more than a minute, say like 3-4 minutes to scan the whole germinal center. Then once you go down again, you can't know which cell is which anymore because they're moving so fast. Okay. So they made little movies just of the border between the light zone and the dark zone, and they saw, oh, look at this. Some cells cross the border in one direction, some cross in another direction. And that was the first evidence, real evidence that cyclic re-entry was happening. But it wasn't very good because, the numbers of cells that crossed were very few and statistics were a nightmare. And then you would expect to see many more cells going up than cells coming down, which also they didn't see in these early movies. They just saw a few cells going up, a few going down. So they weren't sure about this. So what we had to do to overcome this problem was what I did in this paper that we're discussing was I generated this mouse using a protein that is a photoactivatable form of GFP. And this is a really cool protein that was generated by Jennifer Lippincott-Schwartz at the NIH. And it's a form of green fluorescent protein that's usually invisible under our normal circumstances unless you previously hit it with a laser that's of a little bit different wavelength, and upon which it becomes green and stays green. So, in effect, this is a way to label cells in vivo and exactly at the place where you want. So what I did was I took this germinal center, I took cells in the dark zone, and I labeled them green using this system, and then I just took a picture of the germinal center every hour for 6, 7, 8, 10 hours. And all I had to do was, instead of tracking individual cells crossing the border, I just counted them on the other zone. So that allowed us to get some really nice, precise estimates of how many cells come from the dark zone to the light zone and how many cells go back.
Right, because now you can tag them and watch them.
We can tag and watch. We call it geotagging. So we can geotag the cells and then see where they are later on.
So those guys are green, and one of the ways you differentiate the zones is that you've labeled the follicular dendritic cells red, right?
Right.
So it's a very clear, the pictures are gorgeous.
Yeah.
Very clear delineation.
So you can do whatever shape you want with this. I don't know if Vincent showed you some of the pictures, but I'll try to put one on the episode.
Yes.
You can actually draw whatever you want inside a lymph node with the system. So it's very precise.
Well, you've got the GF. Yeah, we got this one that looks like an antibody, and you've got the GFP thing in your paper.
Right. So it actually turned out very nice. This protein's really amazing. This photoactivatable GFP. And now what happened is we labeled either cells in the dark zone and watched them go to the light zone, or labeled them in the light zone and watched them go to the dark zone, and we found that they go at different rates. So the cells that go from the dark to the light zone go at about 50% of them move from one zone to the other in something like 3 to 4 hours. Whereas the movement from the light to the dark zone happens much slower. So we collaborated with a mathematician in Germany. He's actually a germinal center mathematician. So he has these
not over specialized.
No. He has these really cool math models of germinal centers where all the cells are accounted for and they move like a video game. It's really neat. And he calculated that at these rates that we found, that indicates that about 30% of cells that go up will come down. Meaning 70% of the cells
Die, right?
either die or get exported as plasma cells.
Yeah. They're finished.
Now,
right?
these numbers are a little bit sort of rough because of difficulties in the measuring system, but at least they give us an idea.
So
how
70%
stringent-
have either graduated or gotten fed to the macrophages
Right. And 30% go back for
more training.
summer school. Yeah.
Yeah.
So
Not all of you will be moving up. Yeah.
now another question that was in the field for a long time was, are these cells really different from each other, the cells that live in the dark and the light zone? Traditionally, histologists look at sections and then they would call them centroblasts and centrocytes. The centroblasts are the cells that are in the dark zone and they are dividing. The centrocytes are small cells that are resting in the light zone. And we were wondering, when we started to look at germinal centers in vivo, the cells looked exactly the same. You couldn't distinguish a centroblast from a centrocyte in vivo imaging. They looked exactly the same. They behaved exactly the same. And what we wanted to know is, well, how different are these cells? And the way that immunologists look at different populations of cells is that they find antibodies that stick to them, and monoclonal antibodies. So this is a technology thing. It's nothing to do with what we're talking about here with antibodies. But they find antibodies that can label a type of a population and antibodies that can label another one. They mix the cells with these antibodies in different fluorescent colors, and they run it through a flow cytometer. So, for example, you know that there are CD4 and CD8 T cells that are important for different things. And one way of distinguishing them very easily is you put an antibody against mouse CD4 and an antibody against mouse CD8 together with these cells. And they're labeled in different colors. You run them through the cytometer. The cytometer counts every individual cell and says, is this a CD4 cell or a CD8 cell? And gives you the numbers. And then you can use the cytometer to sort these cells into different buckets and then extract RNA from them, for example, and look at what genes they're expressing. Now, this was never possible with centrocytes or centroblasts because we never had markers for them. We didn't know, centroblasts express CDX and centrocytes express CDY. Okay, we didn't know how to do that. So what I thought is maybe I could use this photoactivation method to find that out too. And what I did was I painted cells green in the dark zone, or I painted cells green in the light zone, and I smashed these lymph nodes and put them in the flow cytometer and said, okay, now sort for me the cells that I've painted in one zone, or sort the cells that I've painted in the other. Then we got RNA out from them, and we said, well, let's see what the gene expression differences are. And we found that this molecule CD83 is expressed highly in the centrocytes or the light zone cells and not in the dark zone cells. Now, Jason's sister, who's an immunologist at UCSF, did fantastic work showing that this chemokine receptor CXCR4, which is also an HIV receptor, is what takes cells down to the dark zone. So without that receptor, you don't get the dark zone light zone polarization. And he postulated that was expressed highly in the dark zone. And we actually found that was true. We could see that dark zone cells express more CXCR4 and less CD83, and the light zone cells express more CD83 and less CXCR4. And now we put them in a flow cytometer without photoactivating, and we can separate them. And now we know how to identify these cells in vivo. So this photoactivation also helped us to define markers for these populations.
Is that differentiation good enough so that if you can you label the lymph node with antibodies to those markers and distinguish the zones?
We can, and that's what we do in our work. And
okay,
yeah, so they're not super separate. They're not like CD4 and CD8 cells, but they're separate enough that we can identify them.
But so you can do that on a live mouse. You can infuse antibody and stain, or you have to take out the lymph node to do that. So if you wanted can you use 2-photon to stain with antibodies to different cell types?
To a limited extent, yes. We could. Before we had fluorescent antigens to label FDCs, we used to label them with an anti-FDC antibody.
Okay.
So to a limited extent, you can do that. And so now we had these populations. We knew that they were different. We had a flow cytometry assay for this. And we also knew what the rates were from going up and going down. And we decided to ask the real biological question. So what is telling these cells to go up or go down? And there were two possibilities. One, that these cells would go there. The B cells would hit the FTCs with antigen, and that would signal the cells, okay, you're a good cell, go back down. And the T cells would be there in some undefined role, such as providing growth factors or allowing the germinal center cells to survive after they were selected or something like that. The other possibility was that the T cells were actually doing the selection. So the B cell would go up, it would stick to antigen on an FTC, it would get some survival signals. But it doesn't matter if the T cell sticks very well or very poorly, it still gets the signals. What matters is that if it sticks well, it's going to eat a lot of antigen. And if it eats a lot of antigen, it's going to present a lot of antigen to T cells. So you're mapping antibody affinity onto the amount of antigen that the B cell is presenting. So good B cells acquire a lot of antigen and present a lot of antigen. Bad B cells acquire a little bit and present a little bit. And then it's up to the T cell to choose which one of the B cells to help and to have progress. And to cut a long story very short, we found a system where we could force the B cells to present a lot independently of how much affinity they had by targeting antigen to them externally using antibodies to this molecule called DEC-205. And it turns out that if you do that, the B cells very readily migrate down from the light zone to the dark zone and start to divide. And this happens within a few days. And you can force the selection of a certain group of cells just by forcing them to talk to T cells more than another group of cells, regardless of their affinity. So this is the final. I think the final part of this work was that we showed that if a B cell has the ability to talk to a T cell, that's enough to get this T cell selected. And we believe that this is the mechanism by which B cells are selected within the germinal center.
So it's not what you know, it's who you know.
Exactly.
Great. Fantastic.
So these are T helper cells.
They're TCD4 helper cells. They're a subset that's now very much in vogue, which is called the T follicular helper cell inside the germinal centers.
So, what does when the B cell has the right amount of antigen? What signal does the T helper cell supply that allows it to survive and go on?
A classical signal that T helper cells give to B cells is ligation of CD40. So there's this molecule CD40 that is on the B cell and that activates NF-kappa B.
Which is a transcriptional activator.
Exactly.
And turns on a lot of genes that are needed.
We're very interested in what the signals are that are really doing this. And we also, I think, need to acquire a lot more data on this selection process to be sure whether it's really T cell dependent selection, or whether the BCR affinity, the signal that the cell gets from the antigen itself, how much of a role that's playing. We know that that's essential and that's very important for the cell to survive, but we don't know what exactly is its role in the process of selection.
So you don't know the signals that are turned on by the say ligation of CD40. You don't know the specific genes that are involved yet, right?
There are some candidates, but
is that something you're going to work on?
Hopefully.
Hopefully.
We're thinking of systems to address that.
It's amazing. So do we know how many cycles a B cell will go through?
No, that's a very interesting question because it varies very much depending on what kind of response is induced. So if you induce the kind of responses we do, which is with soluble antigen, that you get a very short germinal center that lasts for about a week or so. Now, if you immunize with antigen and adjuvant, you get germinal centers that last for maybe 3 or 4 weeks. If you infect mice with viruses, you can get very, very long-lived germinal centers. And we think that, for example, HIV in humans leads to extremely long germinal centers that maybe don't even ever go away. Because when you look at human HIV antibodies, especially the ones that are very good at neutralizing HIV, which is an interest in our lab, and the Nussweig lab at Rockefeller, these antibodies have hundreds of mutations, whereas the ones we induce by immunization have maybe 10 to 20 mutations. And why is it that germinal centers last forever, and is that necessary to make good neutralizing antibodies for HIV is, I think, a very interesting question.
Yeah, I've heard that story that the best neutralizing antibodies have a lot of mutations. These are somatic or, I'm sorry, you just said it, but somatic or germline?
Somatic.
Somatic mutations.
Yeah.
I think it's a similar story for broadly neutralizing influenza antibodies. I don't know if you're familiar with that.
I'm not familiar with that story.
They have a lot of somatic mutations as well.
Okay.
So I don't know why. Maybe it's just coincidence that in two different systems, the broadly neutralizing antibodies are highly somatically mutated. If it's coincidence or if it's some functional underpinning to that.
And I think that's something we're going to have to tackle if we want to really generate these vaccines that lead to broadly neutralizing antibodies.
Right.
You said at the end of this paper, which is just, for those listeners who can tackle it, it will take you a long time to get through because there are just so many reagents and approaches, but it's really an amazing combination. You say, this combination of photoactivatable fluorescent protein and two photon microscopy flow cytometry, this combination can be used to other problems in biology. Can you name one of them, just to give us an idea? What else can be done? Be studied?
If we stick to immunology, for example, the dendritic cells that leave the tissue and go into the lymph nodes, those can be easily labeled by photoactivation. We've done that a few times, and you can see them moving around. Immune cells
and all of developmental biology.
Right. The trick will be to get the microscope into the womb and onto the fetus. But
I'm thinking of species where you have access to
maybe in zebrafish.
Maybe in zebrafish you have a clear embryo, but it's still difficult to track cells place to place during particular stages, and you can use this sort of thing.
Exactly. It's. I think it can be. It's gonna. I've had requests from people that do neuroscience or other areas to send them this mice, and we've sent it to many places already. And I wouldn't be surprised if somebody comes up with a neat idea in another field.
Well, sure. It's a natural selection,
Yeah.
Yep.
Put a lot of smart people on a problem, you get neat ideas. So are you going to have this microscope in your lab at MIT?
Yeah, we're just getting it.
You're buying
installed in January.
And you have to have a person to actually run it. You can't do it yourself.
I can run it myself. Yeah.
So you will be doing that.
Right. We need of course you need specialized maintenance to come and tweak it when something's wrong.
So when you publish your first paper, will you come back and talk about it on TWiV?
Sure. I don't need to come back. I can talk over.
Oh, that's right, on Skype. I mean that. Yeah.
How much does one of these scopes cost?
Something like $500,000.
Okay.
You're not going to be buying one, Rich.
It's going to depend on.
I'm just curious.
It's going to depend on what features you have on it and how much you're willing to go bare bones.
That's not really much more expensive than a confocal scope.
If you're willing to go really bare bones, the expensive thing is the laser. The microscope is very simple. Mm-hmm. The laser is a 2-photon laser, which is actually super high technology and costs a lot of money.
Amazing.
But the resolution is just a cell. You can't go down to viral resolution, right?
You can go down to subcellular, maybe, the vacuoles and things like that.
So do you have a number for in nanometers offhand?
It varies so much depending on the tissue, because if the 2-photon effect happens at the focal point, and the size of the focal point varies depending on how deep you are in the tissue. So it's very tricky to. Because
some of these large viruses, a few hundred nanometers may be.
People have done viruses, but I don't think they see virions. They see just deposition of virus in tissue.
Very cool. Anyway, it's beautiful work.
Thank you.
Thanks for talking.
Yeah, it's very nice.
Awesome
stuff.
Wonderful.
Great. Yeah. And again, listeners, check out this paper. I don't know, so they're not going to. Those of you without access aren't going to be able to see.
No,
it.
It's free on PubMed Central.
It's on.
Wonderful.
The manuscript is on PubMed Central.
And also, you have some great photos on your website. We'll post a link to that.
There's a macrophage eating the failed
B cell.
Yeah.
Yeah, we ought to use one of those for the episode image.
That's really cool. Yeah, so check those out, and I'm sure you'll have more of those over the coming years. And you make movies as well as the moving cells, right?
Right.
Yeah. So
the movies on my website don't work. I've tried to make the movies play on different browsers, and they
don't work.
They don't. So don't be disappointed.
But you can access some of the movies at the Cell site, right?
Right, at the Cell site.
Well, you'll figure that out. You got some IT people up there that can help you, right?
Yeah.
All right, let's do a couple of email. The first one is from Neil FWS on Twitter, who sent a link to an article, and he said, "What do we think about this?" The article is "Immunize or Lose Benefits, Parents Told." It's from it's an Australian story.
Right.
And basically, parents who do not have their children fully immunized will be stripped of family tax benefits under a scheme announced by the federal government. So the government says 11% of 5-year-olds are not immunized in Australia, so they're going to take away tax benefits. So what do we think about this?
I think it is an excellent idea.
Yeah?
I agree.
And I think it's I think it's actually better in a lot of ways than some sort of policy where the government says thou shalt absolutely do this, or we'll throw you in jail or something. It's hitting people in the pocketbook, but the reason that's appropriate is these people are imposing costs on the rest of society.
Mm-hmm.
They're relying on herd immunity to protect their kids, but they are doing nothing to participate in that herd immunity. So they're effectively shifting the cost of protecting their kids against these vaccine preventable diseases onto the rest of us. I mean, the rest of Australians in this case, but people who don't vaccinate their kids are pushing this onto somebody else, and I think it's totally appropriate to bill them for that.
Mm-hmm.
I was thinking it would. What do you think about the idea of having something a little more specific to the vaccination? Like if your child gets a disease because they were not vaccinated, then your insurance doesn't cover it.
That then punishes the kid, which I don't think would be fair.
Okay.
How does it punish the kid?
Well, because the kid might need treatment.
Oh, so the parents would just not do it then?
Right.
Yeah,
I see.
something like that.
Yeah.
Not only that, but it maybe doesn't cast quite as broad a net.
Yeah, and then that only kicks in.
People might be willing to roll the dice on that one.
Exactly. It only kicks in if something bad happens. Whereas this, it's, look, by not immunizing, whether anything bad happens to you or not, you're freeloading. So stop freeloading. We're going to take away the tax benefits.
Gabriel, you agree with that too?
I do. I think it's a little bit like secondhand smoking. It's secondhand non-immunization.
Yeah.
That you have the right to your own behavior as long as it doesn't affect others. And I think not vaccinating your children is definitely affecting other people.
Yeah. Could you imagine this happening in the U.S.?
What a stir! Why? What an incredible stir this would make. I mean, after all, the healthcare law where we're requiring people to buy health insurance is going to the Supreme Court.
Yeah.
Right.
Yeah, I have doubts, especially in an election year, that this would ever happen in the U.S., right?
Oh, it definitely would not happen in an election year.
So, okay, thanks for that, Neil. That's a very interesting article. Another one is from Raphael, who is our friend in the Philippines. He's a physician there. He said, would like to give you a heads up on an article I recently read with this link. Gardasil, HPV-4, and the H1N1 vaccines giving a high risk of miscarriages. Would you be able to comment on this? Thank you very much.
So this comes from the National Vaccine Information Center, which, despite the fancy name and the fancy banner, we should all understand is essentially an anti-vaccine organization.
Mm-hmm.
They are neither national nor an information center.
Correct.
As a matter of fact, in many ways, they are a disinformation center.
Yes.
And this is an article where somebody has done, based on some rumors that there might be increased miscarriages with the H1N1 flu vaccine, they have decided to query a thing called the VAERS, which is the Vaccine Adverse Effects
reporting system.
Reporting system. Okay, so this is a federally mandated system where anybody who wants to report some sort of thing that they think is a vaccine adverse effect can send it into this site, and they get about 20 to 30,000 reports a year that are largely unscreened. And it's very important to understand that there's, as we say in the trade, no denominator here. In other words, you're seeing you get a report of something that could or that might or might not be associated with a vaccine, but you don't know what the frequency of that is amongst all of the people that were vaccinated. It's just a report, and it's just a correlation. It's an association, probably in time. Something happened after this person had a vaccine. So there's really not a. It's a canary in the mine, really. It's there so that if there is some signal that comes up that's absolutely overwhelming. You can recognize it and then do further research to verify.
And it also provides raw data if you get a signal from somewhere else. For example, in an ongoing surveillance program, you see, hey, you can closely monitor some small sample and say, well, in this small sample that we're closely monitoring, we're seeing a slightly increased rate of such and such. Then you can go to VAERS and say, what are all the reports of that? Let's investigate.
Right. So what this individual did was to go through this database, and it's publicly accessible. Anybody can go into it and screen it, look for whatever they want, and selected for parameters that had to do with abortion or stillbirth events, okay? And then made a kind of a graph that reports the number of these associated with all of the different vaccines. And the graph that you get out looks impressive because it's got big peaks on it associated with a couple of different vaccines. One is the influenza vaccine. The other is the Gardasil, the papillomavirus vaccine, where they're reporting 200 to 300 abortion or stillbirth events by the vaccine. What they don't tell you is that is for the population a normal frequency of stillbirth events.
Right, because you're not looking at the denominator. This is not a rate. Right. This is just the number of events.
Right. And it's deceptive, among other things, because these are signals above what looks like a background of very few with these other vaccines. But the vast majority of these vaccines are given to children, and children don't often have abortion. Have aborted pregnancies. Last time I saw a pregnant 2-year-old, I don't think I've ever seen one. Okay?
It doesn't occur commonly.
It occurs to me that one of the reasons there might be a signal that an apparent signal that's higher with HPV4, or let's take the H1N1, right? That was in a pandemic year. A lot of people got that vaccine all of a sudden,
and it was aggressively marketed to pregnant women.
Okay,
and who's going to have miscarriages? Pregnant women.
And HPV4 is a relatively new vaccine as well. For all I know, this has to do with expanding the usage of that to a population that would include pregnant women. Okay.
Yeah.
So the bottom line, and then he goes through a bunch of phony calculations and concludes that the H1N1 flu vaccine is 60 times more likely to cause miscarriage than the seasonal flu vaccine. That is completely bogus. All right? You can't do the calculation. That's totally false. Okay? So what's the motivation? What's the motivation?
Yeah, that's what I want to know. What is this guy? He wrote this article.
He's either he's not stupid. Stupid is when you are incapable of understanding something. He's either ignorant, okay, because he doesn't know how to use this stuff, or it's actually he's deliberately putting out misinformation.
Well, there's a third
he doesn't like vaccines.
There's a third possibility that I think actually covers this situation, which is delusional. And that's it's not consciously deliberate. It's somebody has gotten into a non-reality based frame of reference and is now spinning all facts that they see to match that non-reality based frame of reference. And that is what you see in most of the anti-vaccination world.
Okay.
And by the way, when I first saw this link come up, a couple of weeks ago we started talking about this, and I went to the article, and the first thing I do whenever I'm fact checking something like this is I follow the links. So if you see something like this, you say, okay, first sentence, it has been suggested the H1N1 flu vaccine causes miscarriages. See, for example, here, here, and here. And I followed the links, and the first one is actually a press release about a scientific article that makes precisely the opposite claim. Okay, it's a report in the Lancet that found no increase in any kind of event like this in response to that vaccine. The second one is something on a natural health type of blog, which is just a non-peer-reviewed collection of statistical jiggery-pokery that somebody did, which is it's a lesson in how to lie with statistics, where they manipulate a bunch of numbers, kind of like this blog post does, and try to make an argument. And the third one is this complete hearsay news story. Hundreds of women claim miscarriage after taking vaccine.
Okay.
And so that's, it has been suggested that the vaccine causes miscarriage. No, it hasn't. Nobody has made a credible suggestion of such a thing, and even the data that we have show that it doesn't. So that's before you even get past the first sentence.
This is the organization that put the infomercial on Delta Airlines.
That's right.
Yes.
Exactly. Yeah.
Yes, they're the centerpiece of the they're the mothership for the anti-vaccine movement.
This is just garbage, folks. Raphael, I never look at this organization. It's just all misinformation and nonsense. It's wrong. Don't waste your time. Go to the CDC and they'll tell you none of these had anything to do with these vaccines. They look at each one,
and they'll give you references.
This is just awful, and really, I don't understand why they make such a big deal of it. Why they write blogs, they publicize, they spend money on telling this to people, because it's all wrong. I just don't know the motivation unless they're totally disillusioned.
Yeah, they're delusional. They have this belief system, and they have to keep flogging it on the public because overwhelming evidence keeps coming out that they're wrong, and they just keep repeating the same false claims over and over again in the belief that that'll make them true.
Unbelievable.
But they're killing people.
Yes.
Okay, they're killing people.
Yes.
We ought to prosecute them for manslaughter.
Sure.
Drives me crazy.
Yeah, that's the thing that they don't seem to get, is that people die when they don't get immunized, and they don't seem to mind. They keep pointing out the false stuff, the stuff that's not correct. So, all right, there you go, Raphael. I think we have to do this sort of thing
Absolutely.
When people send in these reports.
Yeah, well, Raphael, you can do something, and that is, take this home. Tell other people.
Well, Raphael's a pediatrician, I believe, in the Philippines.
Okay.
Raphael, you need to know that this is wrong. Both vaccines are safe. They don't cause miscarriages, okay? So
there can be vaccine safety problems, and those are serious issues, but this is not among them, and that's not an organization that can inform you about them.
Right. Alan, would you like to read the next one?
Sure. Matt writes, my name is Matt. I'm 16 and I live in the UK. For the last 2 months, I've been subscribed to TWiV and enjoy listening to what's going on in the world of virology when I'm not revising for my mocks, that's a UK exam, right?
Yeah, gotta be.
I think it's a soccer team.
Wait, wasn't that when World War I started? No.
You got to pass your mocks or you'll apoptose, okay?
That's when you get sent back into the dark zone.
Right.
Anyway, I'm at a point where I'm deciding what my future career will be. Virology has proved very interesting and is a very vast career option with plenty of discoveries to be found. Even Dr. Nathan Wolfe said in a TED Talk, if an intelligent extraterrestrial was taxed with writing the Encyclopedia of Life on our planet, 27 out of 30 of these volumes would be devoted to bacteria and virus, with just a few of the volumes left for plants, fungus, and animals, humans being a footnote. Coming back to my question, as someone who's involved in virology, could you give me any information about the field? For example, what is the salary like? What would a working day consist of? And how would you go through it? This information will help me make a choice for my future. Please get back to me as soon as possible, as I'm very interested. I'll still be listening to the podcast every week to hear the latest news. Yours faithfully.
No, the pay's not very good, is it, Rich?
Well, no. I mean, you're not going to get rich, but I have supported my family on my income alone and raised 3 children and sent them to good schools and live well on the salary that I make. Okay? Yeah. I'm not wealthy, but it's worked out just fine. Okay? So
yeah, I think you can't go in it for the salary, though.
No.
You got to really want to do it.
The point is you're not necessarily going to make a huge sacrifice, okay? You're going to get paid enough. It's a decent living, all right? But you're not in it for the money.
What's a working day? Hey, tell them your working day.
Oh man, this has been just such a great week. What an amazing week. Actually, let me get up I'm in a phase here where I've got time to do experiments. So I'm looking at my calendar. Monday, actually, I met with a speaker in the morning, and then I infected some cells shortly after that. And then there's a big blank space in my calendar. I don't know what that was. There's some processing some email and maybe reviewing a paper or something like that takes me through Tuesday. And then I spent Tuesday afternoon looking at a confocal microscope on some old samples. And then Wednesday all day long, me and Nassim purified a bunch of virus. And I came back Wednesday night and infected some cells for the following day. And then Thursday morning, I worked up this stuff for this immunofluorescence experiment that I did, and then went to a team taught a class for 2 hours, and then went back at the microscope, and I was there until 6:30 last night, and I was there again this morning, and now I'm doing a TWiV. How cool is that?
Sounds good.
Right?
And I got to say that when Rich was taking those confocal pictures, he was so excited. He emailed me one of them, me and Alan, right? And he said, I'm sitting here right next to the microscope. I got my laptop. Look at what I'm doing. So he's excited.
Now, I don't, this is an unusual time for me. I like to spend time in the lab when I can. I go through weeks where what I do is sit in my office and either plan lectures or review papers that people send me, or if I'm lucky, write papers. I spend a lot of time reading and writing science, okay? And I spend a lot of time talking to people in my lab, going over their experiments and stuff. There's coffee in my office every morning with people from my laboratory that is, it's a very casual affair that can range from how's your family to 80% of it is not planned, but it's talking about the experiments and the science. And then, of course, I do some teaching, stand up in front of students and yak at them, right?
Of course, it depends on what point in your career you're at, but let's get Gabriel to tell us, because in immunology, virology, your day is going to be more or less the same. So at your point in your career, Gabriel, what's a day like?
My days lately have been a little bit unusual because I'm finishing up in the lab and moving to a new one. So, but when I was really working a lot for for this paper, for example, some days I would just get up and get my mouse, put it on the microscope, and image it for the whole day.
Do that all day, right?
Yeah. Or, take a mouse to the microscope, paint the germinal center cells for a few hours, and take them to the sorting.
But you loved it, right?
Yeah, it's nice. And then you have, a few hours when the sorting's going on to go and do some mini preps or something, extract DNA from bacteria. Yeah. You're always doing something, and what I like about it is that there are always a lot of people around you. And
Yes, very good point. A lot of people figure that this is an isolated. They see an isolated ivory tower thing. No way. A laboratory is a very, very social environment. Okay, in my laboratory, there's people shouting at each other all day long.
Yeah, you don't know this, you
It's
go
great
somewhere,
stuff.
you ask a question, you ask
Yeah.
somebody else another question. Somebody comes to you with something they don't know, you tell them how to do it.
Yeah, people are always walking in my office, just jabbing about science or whatever. I'm always walking around talking to people. So it's not a solitary thing at all.
And to me, the really great thing is what your work is to have crazy ideas and execute them.
Mm-hmm.
And that's, I think, what a lot of people dream of when they're just doing their thing, whatever.
Yeah.
I hear a lot of people saying, I wish I could be more creative in my work.
You made a very good point early on, Gabriel, about having a variety of things to do. A couple of years ago, I had never done confocal microscopy. I didn't know how to do it, okay? And I learned how to do it. Years before that, I didn't know much about protein purification. We figured out how to do that. When a challenge comes up, you figure it out, you do it, you learn something new.
Exactly.
It's fantastic.
So you're never bored, and you get to do what you want to do. You want to work on some aspect of a virus or an immune response, you can do it. As long as you can raise the money for it, you can do it. That's the aspect of the job I really like, besides everything else we've talked about. I can do what I want to do. Nobody tells me what to do every day.
And also,
at least here in the lab.
I was going to say, Matt, bear in mind that you're 16, you're looking at college, presumably, and if you decide you're going to major in biology or biochemistry or some other molecular biology, a field that's related to virology, then that's an outstanding choice for a university major, no matter what you ultimately decide to go into. Mm-hmm. So if you ultimately decide, well, I thought I wanted to be a virologist, but now I've finished my degree and I would really rather go do something else, you can go anywhere with a science degree.
Like maybe be a writer?
You could be a writer with a science degree, yes.
And I would also suggest, and we've suggested this before, that at your earliest opportunity in your university, go find a lab to work in.
Yes.
Don't wait. You'll be able to find a laboratory. You don't have to take a lot of courses or anything. The students that I have that get the most out of it come to me when they're like freshmen, the minute they walk in the door. Okay, go find a lab to work in, and then you'll find out from being in the laboratory whether this really floats your boat or not. And if it does, great. If it doesn't, great. You found that out.
Yep.
All right, Matt, I hope you're listening. Yeah. There you go. One more email. Rich, can you read this one?
Okay, let me get back on target here. I was looking at my calendar. Anne writes, Dear TWiV community, I wanted to make a contribution regarding episode 145. You read an email from Wendy at University of Iowa regarding the history of cloning technologies. She referenced the 1970s work at the NIH and scientists' self-imposed or externally imposed moratorium on the work until the risks could be investigated. As you mentioned, there was also a moratorium on cloning work in Cambridge, Massachusetts. A few weeks ago, I stumbled on this treasure, a black and white video recording of the Cambridge City Council hearing on the risks of recombinant DNA work, including city council members, scientists from MIT and Harvard, and a representative from the NIH and Cambridge citizens. The MIT library has an edited copy of this video, 30 minutes total, including some narration available on their website as part of one of their 150th anniversary projects. Please post this link for others who may be interested. It is fascinating to me that one scientist tries to defend the ongoing research as very unlikely to be hazardous, while another scientist points out that this new and powerful technology has a huge potential to help but also to harm, still poorly understood. I think it is important for scientists to be able to talk with each other and with the public about the implications of their work, scientific and moral. I am glad to hear you discuss not only the hot new papers, but also the fascinating investigations from the past that got us where we are today. I think this is wonderfully enriching for scientists to understand our heritage, so to speak. Thanks for all your work, Anne. And Anne is a graduate student at Harvard, and she has the link here to this thing, which I've seen before. It's amazing.
Yeah, well, actually, I think it was a pic.
It was a pic. We posted it some time ago,
Yeah.
but it's worth posting
Absolutely.
again.
Yeah, it's a very cool video. It got, I think David Baltimore is in it, Mark Tashney. Yeah,
David Baltimore's sitting. David Baltimore and Wally Gilbert don't say anything, but they're sitting in the front row in the audience. Okay.
Maxine Singer, yeah.
Maxine Singer,
cool.
Mark Tashney talks, and John King talks. And one of the things that I think it's important to point out is that, although I suppose it didn't have to work out this way, all of the threats of gloom and doom, none of them came to pass.
Right.
Okay.
Right.
Turns out to be quite safe, and what we have learned from recombinant, the potential that recombinant DNA technology provided. I mean, almost everything that we're doing today is based on that. I mean, it's incredible.
Yeah, it's similar to what we talked about last week. It's good when scientists police themselves
Yes.
rather than someone else. It looks good, and if it works out well, everybody's happy.
And we we get it right eventually.
Right.
Yep.
All right, that brings us to Pics of the Week. Gabriel, you want to pick something for us?
I actually have a pick, but I don't know if you want to have a whole show on it, which you mentioned, which is that David Baltimore paper on
that's a good idea. We could pick it and have people read it.
engineering immunity to HIV using a virus. Because what happens is we're not really good at eliciting broadly neutralizing antibodies to HIV with vaccines, and this is one thing we need to learn how to do. But in the meanwhile, what they did was they put a really good antibody into a virus, into an adenovirus, and injected mice with this adenovirus, and the mice start producing the antibody for a very long time, apparently for the whole lifetime of the mouse, and then they are protected against, humanized mice are protected against infection with different strains of HIV. And it's
Very interesting.
Very interesting. It's a very nice discussion of would this ever be actually done in people, and I think it's surely something worth discussing.
Yeah, we have had 4 or 5 requests from listeners to do that paper. Unfortunately, it won't be till next year because the rest of this year's TWiV schedule is pretty booked. But we will do it first thing in January. The results will be the same, even though we do them in January, I assure everyone.
Great.
Thank you, Gabriel.
Welcome.
Rich.
Okay, I'm on a movie kick. This is last time I did Gattaca, and in fact, it turns out this is the same year. The movie Contact. So this is about, actually it was based on a book by Carl Sagan, and it's about contact with extraterrestrials. What I really like about it is that Jodie Foster plays this really hard-ass scientist who absolutely won't buy into anything that isn't based absolutely entirely on observable data. And then through the contact with the extraterrestrials, she realizes that, well, maybe there's another way to look at things. Okay, so at any rate, I like the movie. It's a good movie.
You've seen it, I presume.
I have seen it. Have you seen it?
I have not, no.
I keep finding stuff you haven't seen. I figured this would all be obvious. Have you seen Contagion yet?
I haven't seen Contagion yet.
Okay,
no.
Vincent.
I know, but it's not in theaters anymore, right?
Yeah, you're just going to have to kind of lay back a little bit, take some time, watch some movies. It's okay.
I would have seen Contagion if my kids had wanted to. They're not interested. So, and my wife doesn't want to go. So I feel going by myself to a theater is a bit weird.
Well, I've been that weird occasionally.
Although not really weird. It's okay. I just don't want to make the time to do it.
Right.
But
I kind of had the same problem. I did not get around to seeing Contagion when it was in the theaters because nobody else wanted to see it.
So it's not in theaters anymore. Is that correct, Alan?
Not around here that I know of.
I think that's right. Yeah. So you guys are going to have to get it. We still have to do this.
We have to do a show. I got a bunch of emails from people who have great comments. I agree.
Okay, so go see Contact. That's good, too.
All right. Thank you, Rich. Alan.
My pick is a short clip from a TV news station out in California, and I wanted to highlight this for two reasons. First of all, I don't know if I'm sure a number of our listeners heard this thing that went around last October about this former cheerleader who apparently got all sorts of muscle problems after getting a flu shot, and she couldn't walk and had trouble talking, and blamed the flu shot for causing all these symptoms. Well, surprise, surprise, it turns out she was full of it. Or so it appears. But the effect of that particular video that went viral on the internet last year was a lot of people really questioned whether they ought to get a flu shot. So one of the many news stations that had picked this up was this CBS affiliate out in Los Angeles, KCAL9. So I wanted to highlight this video, first of all, because it goes and reinvestigates that case and shows that was bogus. That should not have been a concern. But also because the news team actually went back and reevaluated that. This is a great thing to do in journalism, to go back to an old story that you covered. Looking back at it, hey, a year ago we had that story. What's happening with that? So what they did was they sent a news team out to find this girl, and she didn't return their calls, and so they went and they tracked her down and sat outside her house when she wouldn't answer the door, and they saw her coming out of her house, walking just fine, everything's okay. They tracked her down as she was at the shopping mall and walked up to her. She's walking fine, everything's okay. And then she starts showing symptoms again as soon as she sees the TV crew. So it's the type of thing that I really wish more journalists would do, particularly for stories like this that are highly inflammatory at the time and news of the moment. And it's really good to go back to that stuff later and say, "Gee, how did that all turn out?" And wow. So thank you to KCAL9, if anybody from there is listening to TWiV. Nice job, nice reporting, and wow. And by the way, that turned out to be bogus.
Wow.
Can we prosecute her?
What is she doing it for? Ridiculous.
Attention.
She got a lot of attention.
Yeah.
Well, she could just make a creative video and put it on YouTube, and she'd get a lot of attention.
Yeah, and now of course she still claims that this is the case, and you can't prove that that's not. But you can go and watch this news clip and decide for yourself.
All right, thank you, Alan. My pick is two websites. These both deal with crowdsourcing research, scientific research. One of them is called sciflies.org. Make science happen. If you want to do an experiment, you're in a lab and you need a bit of money to do it. Maybe you want to buy a confocal microscope. You put it up here and people donate money to it. And the other one is called Rocket Hub, rockethub.com. And again, the same thing. You put up an idea for an experiment and you can crowdsource it. Now, most of these are smaller projects in the thousands of dollars. But you get the money. People donate $10, $20. You get a lot of people doing it. You can do some experiments. So this is kind of a new thing, and I've gotten a bunch of email from people who have their projects and they want them publicized and so forth. There was one here to do an experiment on parasites, for example, for a few $1,000s. So check these out. These are very interesting. I did look to see if they would support education projects, but they just want to do research projects.
Well, there are other sites for education projects.
Is that right?
Yeah.
Maybe you should send me a couple because,
Well, actually, one that I picked a few weeks ago for there was a microscope funding drive.
Right.
Yeah, the site on which those were based does a whole slew of that sort of thing.
All right. I'm looking into ways to support TWiV and the other podcasts,
This whole crowd thing is a new phenomenon. It's another outgrowth of the internet. It's terrific.
It is.
Yeah.
Yep. It's pretty cool. I like it. Got to embrace it. Okay, and we have 2 listener picks this week. The first is from Neva. And I just have to say, look at this. Where did the photo go? It's not here right now. Is anyone seeing this? It's not loading anyway. Anyway, it's a drawing of a virus and a retrovirus.
It's loading here, yeah.
It's just great. And someone had a comment, that's not a virus, it's a bacteriophage. Excuse me, they are viruses. Wow. Anyway, that's a beautiful cartoon. Check it out. Thanks, Neva. That's great. I loved it. I still do.
Yeah.
Our second pick is from Aisha, who says, honorable TWiVnesses. Wow, that's
I like that.
That's good. I heard this series and thought you might dig it for Listener Pick of the Week. The series is in progress, but interviews 1 to 4 are available on the BBC iPlayer or for download as a podcast. Professor Al-Khalili is a physicist, but you have science podcasting and science outreach in common with him. The interview with Paul Nurse is a really interesting window on his work, mind, even personal life. So these are BBC interviews that are online.
Terrific. And, that's our other writer who was that the 16-year-old student might like those.
Yeah. Said right. Paul Nurse. You know Paul Nurse, Gabriel, right?
Yeah, he was our president at Rockefeller until
right.
a few months ago.
So thank you, Neva and Aisha, and that will do it for TWiV 161. You can find TWiV on iTunes, at the Zune Marketplace, at microbeworld.org, or at twiv.tv, where we keep all the show notes and links. There's also an app for your Android device or iPhone at microbeworld.org/app. And don't forget facebook.com/thisweekinvirology. As always, send us your questions and comments to TWiV at twiv.tv. Gabriel Victora is at Rockefeller University, but within a week or 2 will be at the Whitehead Institute at MIT. Thank you for coming back, Gabriel.
It's my pleasure.
It was great. I love it. Our first really in-depth immunology on TWiV, right?
That was good.
Yeah.
So if you like that, we can do some more, because I loved it. I thought it was great. Your work's beautiful. Best of luck in your own lab.
Thank you.
You're welcome.
Yes.
Alan Dove is at alandove.com. Thank you, Alan.
Thank you. Always a pleasure.
And Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Thanks to you, Gabriel, for coming on the show. This has been terrific.
It's been great, yeah.
You're exhausted?
Yeah, my brain is full.
Wait till I get really started on T cells.
You're going to do T cells? Is that
no,
right?
I can't. You need to get a T cell person.
Yeah, you're right.
B cells are simple.
You know,
we've done a lot of innate immunity. We've done B cells. We need to do T cells. You're absolutely right.
Yeah, we'll get a T cell person. But we have to have someone who is as eloquent as Gabriel.
No.
Yeah, nice job.
Great.
Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.